Alright, let's talk about another class of biological macromolecules. You've probably heard of this class, proteins. So the monomer of proteins, they are called amino acids. And the polymer, you can call them proteins. You can also call them polypeptides. Alright, and let's talk about what an amino acid is. So an amino acid is composed of an amine group, which is NH2. It has an alpha carbon in the middle. The alpha just denotes its position. It has a carboxyl group on the other side, which is why it's an acid. It has another hydrogen, and it's connected to something we call an R group. So the R group could just be anything. And an R group is what differentiates one amino acid from another. And in biological systems, there's plenty of different types of amino acids. I believe, there's 20 different naturally occurring amino acids. And let's talk about how these amino acids bond together. So what happens is, let's have two amino acids. And we're going to circle the ones that are going to bond together. So we're going to circle this and this. And now I'll copy it over so we can have a side-by-side comparison. So these two, the H and then OH, combine, you guessed it, it forms water. And the rest of this, it's not an oxygen bridge. It's not the same thing you see in glycosidic bonds or just in lipids. What happens here is that you have an H, you have an H here, and you have an O here, and the result is you have the carbon connected to the nitrogen of a different amino acid, and this allows you to combine amino acids together while forming a water. So it's still dehydration synthesis, and we call this bond a peptide bond. All right, and then let's talk about how these amino acids form protein structures. So there's 4 levels of protein structures. The first is primary structure. And that's just the sequence of amino acids. So it could just be like, let's just have like blue, red, and green amino acids. Although, this is a drastic oversimplification. Like, we could have this, and this would be the primary structure, because that's just a sequence of amino acids. And then for secondary structure refers to amino acids interacting with other amino acids not directly bonded to it. And you see secondary structures in 2 forms. The first form is this. It's called an alpha helix. The second one is this. And this is a beta pleated sheet. And the reason why these secondary structures exist is because of hydrogen bonding. Now, for those of you who need a refresher, hydrogen bonding is the weak interaction between hydrogens and oxygens. It's not an actual bond. They just weakly interact because since oxygens are more electronegative, they're partially negative while hydrogens are partially positive, and that results in a bit of a attraction between the two. It's far weaker than covalent or ionic bonds, but it interacts enough for you to have these sort of structures form. So we can say have hydrogens and oxygens here, and they could be interacting and forming the structure. And then we have tertiary structure. So tertiary, and this is how a protein folds in on itself. So let's say we have a long polypeptide chain. It could fold in on itself. Yeah, and this is caused by interactions between the amino acid R groups. So these amino acid R groups, they could be, polar, nonpolar. They could be ionic, acidic, basic. And that really just influences the tertiary structure, and, it makes it fold in on itself in a specific way. And lastly, we have quaternary structure, and quaternary structure isn't really applicable to all polypeptides because quaternary structures means that there are multiple polypeptide chains folding together, and that's not something that always happens. So let's say we have one chain and another, a third, and this is just how the polypeptide chains interact. So these are polypeptide chain interactions. All right, and then let's talk about the function of proteins, because that's very important. So one use for proteins is structural. Proteins can be used for structural support, and we're not really going to go into that. Far more importantly for us, we're going to explore proteins' role as an enzyme. Bear with me for a second. I'm having some technical difficulties. Enzyme. So what is an enzyme? An enzyme is a biological catalyst. So biological catalyst. And maybe you don't know what a catalyst is. That's fine. A catalyst is a chemical that lowers activation energy. Now this might raise the question, what is activation energy? So if you put a graph of, let's say this is energy, and this is the time. So we have a reaction taking place, and let's say this is an exothermic reaction, so it releases heat. So what's going to happen is these are the products. This is the energy of the products. It has a lot of potential energy, and these are the energy of the reactants. So the reactants are low energy. Now, to have the reaction go forward, you need to have activation energy. Activation energy is just this section. And that activation energy is needed to start the reaction. Now, what an enzyme, or I guess catalyst, would do is it lowers the activation energy. Maybe it does this. In certain chemical processes, you need multiple enzymes to have a reaction take forth. So in some reactions, you might see this, smaller sort of hills, so to speak, of activation energy. And this is really important because a lot of these interactions we see in biological systems, they don't really just go on their own. Either they just don't happen, period, or they just would be way too slow for us living beings. So the crucial role of a biological catalyst, enzymes, is to make sure that these reactions happen. And looking at this graph, you can see that since this is exothermic, the difference between this, so the energy level where you started here, and the energy level you end off with, this is the net energy released. And then we can talk about how an enzyme makes this happen. So there's two models. The older model was called the lock and key model, and it's been disproven. It's not really what happens, but it's a really good model to just like sort of basically understand what happens with an enzyme. So you have, let's say, an enzyme here, and then you have, let's say, something it needs to break down. Let's say this is maltose. Which for those who don't know is just composed of two glucoses. So enzyme. And to break down maltose, you need to have energy to split it apart so that, a human can access the energy. And and by the way, I should probably tell you that the reactant is called a substrate. So what's going to happen is the particular enzyme, in this case, it's I believe it's called salivary amylase. Salivary amylase. It's you can see here. This is not really what happens, but you see, it matches exactly maltose. So the maltose fits perfectly into the enzyme. So it's like you have a lock and a key. They have to fit perfectly. Now let's say we have another enzyme with maltose. This enzyme is shaped a little different. Now you notice that this can't fit in there. And because of that, it can't react. So only the specific enzyme with this shape can react. So that allows specific enzymes to allow specific processes. And then what happens is that the enzyme reduces activation energy. And it's going to break the maltose apart. Well, it's not going to break them apart, but it's going to lower the energy to a sufficient point where other processes can break it apart. So we're going to end up with two glucoses. Now keep in mind that this is like super simplified. This is hardly at all what happens, but this is just an approximate model to help you understand. Now let's talk about the other model. The other model, it's called the induced fit model. So the induced fit model is, well, we call this place the part of the enzyme that's being used. We call that the active site. So in an induced fit model, the enzyme's active site isn't truly a fit. So for the substrate, so maybe it's like this, and the substrate is like this. So it's not quite a fit, but it's close. The idea behind induced fit model is that the enzyme's active site changes shape slightly. So you have the shape here. It might change slightly to something that fits the substrate. And in reality, what happens is you have certain arrangements sort of rearranging itself. So maybe you have like a molecule that was this as part of the active site, but then it rearranges itself to this. Now the molecules, the atoms in the molecule stay exactly the same, but what's happening is that you're rearranging them, so the bonds stay the same, the atoms stay the same. You're just kind of shifting them subtly so that all of a sudden the active site fits. Okay, now that we've gone through how enzymes work, sort of, we can talk about denaturing. So denaturing is when a protein loses function thanks to changes in the environment. So the two most typical sort of changes in the environment you see that cause denaturing is pH changes and temperature changes. And you might ask, how does the protein lose function? Well, it loses function because its structure breaks down. And the structure breaks down in reverse order of the protein structure we just talked about. So it's going to break down quaternary first, if it has a quaternary structure, and then it's going to go down to tertiary, secondary, and finally primary. Now, you might also notice that since you're going from the more all-encompassing levels to the more specific ones, that certain changes can be reversed. So let's say tertiary structure was changed up. This was changed up, and then all of a sudden, let's say the temperature goes back. Now, since it's only the quaternary structure, the protein might actually regain its function because it's just a quaternary structure. It's not something that's super basic, so it might be able to regain its function. Now, on the other hand, let's say the temperature got super out of hand, and let's say the secondary or the primary structure was disrupted. Now, that would likely make it impossible to regain function. Because if you're breaking the primary structure, well, you're just breaking the amino acids up bit by bit. It's not really even a protein anymore, so it's not going to function. And then now let's talk about how proteins are controlled. So proteins are controlled either through positive or negative. So positive control is when something turns the enzyme on. So something turns the enzyme on. While for negative control, something turns the enzyme off. And for positive control, you normally you do it for enzymes that aren't as needed, so not as needed enzymes. And for negative, it's the opposite. You use it for more frequently used enzymes. So a way I would suggest you remember this is think about it like the enzyme is a light bulb. So we have a light bulb over here. And let's say that the light bulb is going to be used a lot. Well then, if it's almost going to be always on, well, you'd think that it stays on the on switch. So it's on unless you tell it otherwise. So only when you flip it to off will the light bulb go dark. If you're not telling it to go off, it's going to stay on. Now let's talk about a light bulb that needs positive control. So a positive control light bulb, well, you're not going to need it a lot. So normally it's going to stay dark. However, when you need it, you can turn it on. Sorry, give me a second. I'm having an issue with my stylus. So it's normally off, but if you want it to turn on, you have to tell it to. So then it becomes on. But the moment you stop telling it to be on, it's going to go back to off. So that's a good way to remember it. Let's talk about how it exerts control. So there's two ways that control can be exerted through. So the first one is just called competitive inhibition, or I guess competitive control. I'm just going to use the word inhibition because we're going to pretend we're just talking about negative control. So competitive inhibition. So how competitive inhibition works is, let's say we have an enzyme and it processes something we just call substance A. Now it's going to break substance A down, or it's going to make it undergo a chemical reaction. But the point is, let's say we can make another thing, a substance B, something that can still fit within the active site on the enzyme, but it doesn't break down. It doesn't react. Well, what's going to happen when we distribute substance B? Well, the end result is going to be that it's going to get clogged with B. It can't do anything about it, so it can't do anything with substance A, so it's blocking the reaction. So and the reason why it's called competitive is because you can think about it as it's competing for the active site. It's trying to compete for it. It's trying to outcompete the other substance. Either one is called non-competitive. So how non-competitive works is that you have the enzyme. You have the enzyme, and then you also have another site, a sort of secondary active site. And this site is called the allosteric site. And the allosteric site, it can't connect to, let's say, substance A again. So substance A can't fit in the allosteric site. So you're not. The inhibitor can't compete with the substance that's being reacted. So what's going to happen is that you're going to have the substance B again. This time it's a non-competitive inhibitor. It's going to go connect to the allosteric site, and that's going to shut down the enzyme. So the enzyme is going to be shut down. There's no competition. No matter how much of substance A you throw at it, it's not going to function. And this leads me to another concept. It's not really like competitive or non-competitive. It's called feedback loop inhibition. So let's say we have a few different enzymes that have different active sites, do different things. Let's say we have enzyme A, enzyme B, and enzyme C. So we have a substance in A, goes through, it fits in B, goes through, fits in C. And now the end product, let's say, is an inhibitor of A. So your body uses this because it's decided, oh, I have enough of this substance. Let's call this substance D. Substance D turns out to be an inhibitor. It stops the cycle. Stops the cycle. And that means there's no more production. No more production eventually leads to, well, no more inhibitor. That's going to result in the process starting up again. So you see how it's a feedback loop. It controls itself. It's a cycle that can maintain itself. And then lastly, we can talk about sort of the effects and how competitive inhibitors can be overwhelmed. So I'll draw this graph. So this is going to be the rate of reaction versus how much substrate we're going to put in. So without inhibitors, it's going to look something like this. And it caps off at a point. Because you have only so many enzymes. So this is the max amount. Now let's say we have competitive inhibitors. We introduce competitive inhibitors. Well, then what's going to happen is that you're going to have a slower reaction rate because you're competing, but eventually it's also going to go to max because eventually you can just stuff such an amount of substrate that no matter your competitive inhibitors, they're going to get overwhelmed. Then lastly, we have non-competitive. And that's just going to lower the rate of reaction to a certain amount because you can't overwhelm non-competitive inhibitors. Now, the maximum that it can react, so this point over here, this line, is called Vmax. And then there's another measurement called Km. So Km is half of Vmax. So for the reaction without inhibitors, this would be Km. So that dotted line would be Km. And the reason why Km is so important is it shows us how easily enzyme can be saturated or go to Vmax. Because if you notice here, the slope becomes lower and lower. And that's because Km is the first half. So at Km, you can saturate it really efficiently. So this is very efficient in terms of how well you can saturate. But after that, you can see the slope kind of just curves out. So that means it's less efficient. So Km just tells you how efficient it is.